Finally. It's about 12 hours to get there. That technology was supposed to make things faster and more efficient, not slower and more lethargic. That look about right? Okay. So we're talking about extensions to Mendel. And we started talking about polygenic inheritance. Basically in polygenic inheritance, what you've got is a violation of A, right? So remember Mendel's trait pattern that he saw was one gene, one trait. This is a violation of A. So in this case, you have many genes for one particular trait. And as a result, oftentimes when you have multiple genes involved, what happens is it kind of smooths things out. So like, for instance, if you were to take a look at a trait with just like 3 potential outcomes, right? Tall, short, and between. Then what you would get is you would get one short, in between, and tall, right? Basically 3 buckets. Now, the more genes you add, then the more variations in between those 3 you get, right? So if you have more variables, then what happens is you don't just have short, you have, well, maybe not so short, and then not little short, little less, and then same thing with the tallness, right? So you kind of have all these like variations in between, and then maybe you get super tall and super short. So the more variables you add, then the more data points you get in between that. Now, if you connect all the tops of these, which you'll see, is something that looks like that, which is what? Yeah, normal distribution, right? Basically the bell curve, which is also referred to as a continuous distribution. So this is the reason why polygenic traits are oftentimes referred to as continuous traits, or sometimes quantitative traits as well. And typically this kind of bell curve shape is kind of a dead giveaway for a system where there's multiple genes involved in there, not just one. So a good example, for instance, is height, right? So if you basically have everybody sort of line up according to height, then basically what you're going to get is you're going to get really, really short people, but not very many. Really, really tall people, but not very many. And most people are going to be somewhere in between. So you're going to get a natural bell curve, a continuous trait. Because you don't just have 4-foot people, 5-foot people, 6-foot people, right? You've got people that are 4'1", 4'2", 4'3", 4'4", 4'4 1/4, 4'4 1/2, 4'4 3/4", right? You have every single point in the continuous line, right? And that's what creates this bell curve. And there's several traits like that. So height is one of them. Another common one that's in some textbooks, and in other textbooks they avoid it, but it's actually really good, is skin color. Your skin color has actually got something on the order of, I believe, if I recall correctly, something like 8 genes involved in it. And all the genes are the same, right? You have 2 alleles. You have an allele for pigment, dark pigment, right? And then light, right? So not the absence of pigment, that'd be the albinism, right? But you have 2 different alleles, dark allele, light allele. And when you take a look at all 8 of your genes, your skin tone is basically reflective of the accumulation of how many dark or light alleles you get, right? So generally speaking, when you're talking about 8 different genes, to have all dark alleles, remember you're talking 8 genes, 2 alleles, right? So to get all dark alleles is a very rare occurrence. To get all light alleles is a very rare occurrence. So that would be somebody who's very, very light skin tone versus very, very dark skin tone. It's more common to get a mixture somewhere in the middle, right? Which is a reason why when you take a look at skin tone across the world, not just, in a classroom or even a country for that matter, but you have to take a look at the entire world, the entire human species. Skin tone is basically somewhere in the middle. It's kind of an olive-ish color, It's kind of an olivish color, sort of either leaning dark or an olivish color leaning light. It's somewhere in the middle. That's where the majority of the human population is. By the way, this is also one of the reasons why when we sort of took a look at hair color and eye color in the lab, it's a little, if it were up to me, I'd probably remove those. Because eye color and skin color are just pigmentation, as is hair color, right? So your pigmentation typically is governed by essentially the same laws. This is the reason why if you tend to have, it's not exactly 1-to-1, but for the most part, if you have pigment in your skin, you'll have pigment in your hair, right? So basically you have the similar pigment genes, it's just you're depositing it in different areas. But this is also one of the reasons why hair color is a lot more complex than just light and dark, right? And eye color is more complex than just light and dark, is because your pigmentation is not just light or dark, right? Remember when we looked at that list? We looked at that list, what we were assuming was that everything on there was Mendelian, that it obeyed those rules, right? One gene, one trait, classic complete dominance, right? 2 alleles, all that, it obeyed that. But eye color and hair color do not, right? We know that because even without assistance from product, right? We know that there's a lot more to the story of hair color than just light and dark. Even if you took individuals with just dark hair, there's a variation within dark, right? I mean, there's people with like jet black hair, and there's people with kind of dark hair, but it's kind of like got some highlights in it, or it's a little kind of, it's not quite as jet black, okay? And so there's variation even within that. And those are all complexities in genetics that we really dig into, because it's like, well, wait a minute, how does this actually work when we get into the genetics class? So that's kind of the sorts of things that the genetics class likes to get into. So that's polygenic inheritance. The next one is pleiotropy. And pleiotropy is similar. So it's a violation of A. In this case, you have one gene, but this will lead to many traits. So that's a violation of the one gene, one trait component of Mendel, the Mendelian inheritance. Oftentimes where we see this is usually in human diseases. That's usually where we see this. Because typically what happens is, like in a human disease, we'll have one gene that causes the disease, but the disease itself will have multiple different symptoms, right? A good example is cystic fibrosis. Sickle cell anemia is another good one that we see. So when you take a look at these, like cystic fibrosis is a really good example, right? Because cystic fibrosis is essentially a mutation in one gene, and it's a gene for a chloride channel. But the chloride channel affects lots of different systems. So when you take a look at cystic fibrosis, for that one mutation, you're going to get symptoms in the liver, mess ups of your fat of digestion and absorption. You're going to get pancreatic symptoms. You're going to get respiratory symptoms. So your cilia that's in your respiratory tract don't have the ability to flush out the phlegm out of your respiratory tract when it catches particles in there. So you can't actually move it out of there. So it kind of feels like you're drowning all the time. And so your lungs will fill up with fluid. And then your sweat glands, you have an abnormal amount of salt secretion in your sweat glands. So you're losing an abnormal amount of salt. Why? Because it's a chloride channel defect, which makes sense. Your chloride has to go somewhere. And so oftentimes you excrete it as a byproduct in your sweat glands. So these are all different systems. So when you take a look at the symptoms of each system, it's considered separate phenotypes. So these are considered separate phenotypes or traits from a physiological view of things. Okay. But it's all caused by one gene. So this is an example of pleiotropy. So those are violations of A, the one gene, one trait thing. But now let's go ahead and take a look at a violation of the second one. Okay, what happens if your system, your gene, has more than two alleles, right? That's called multi-allelism. So that's a violation of the second one, B. This is where a gene has more than two alleles in the population. Notice what I didn't say. This is important to draw this distinction. I didn't say more than two alleles in an individual. That's not possible, right? You only have two. You got one from mom, you got one from dad. There is no third piece, right? So you only have two, but the population has more. Okay, and we'll take a look at a good example of multi-allelism here in just a second. But before we cover multi-allelism, what I want to do is I want to take a look at the next set, which is a violation of the third criteria of Mendel, which is the complete dominance piece. So multi-allelism actually is best illustrated in the ABO blood grouping. So there's three alleles there in the population. You only have two of them, but there's three in the population. We'll take a look at those. But the ABO blood system is also a really good example of another type of pattern of inheritance called codominance. So these are violations of the third one. All right, what happens if it's not completely dominant? Oftentimes this kind of question comes up. It's like, well, like what happens if the white doesn't get completely masked? Well, that's going to be a violation of Mendel, and that's going to be an alteration in this complete dominance idea. So there's a couple of different versions of this. The first one is incomplete dominance. This is basically where a dominant allele does not completely mask the recessive. So if you can't completely hide the recessive, then you're incompletely dominant. Now, similar but not the same is what's called codominance. Codominance is where two dominant alleles are equally expressed. Notice I underlined equally. So this is a straight down the middle, 50-50 power sharing structure. That's what this is. So you see evidence of both. There is not one that is greater than the other. They're equals, 100% equals. Okay. And so that's what codominance is. Because if they weren't equals, then it would be a version of incomplete dominance, right? So codominance, typically, they're equally expressed. And multi-allelism and codominance are best seen in the ABO blood grouping. But before we get to the ABO blood grouping, which will explain multi-allelism codominance, we want to talk a little bit about incomplete dominance, because we actually already saw incomplete dominance. We just didn't call it that because we weren't ready yet to call it that. Okay, so basically we're going to talk about incomplete dominance first, and then we'll talk about the ABO blood grouping and multi-allelism codominance. So incomplete dominance, where'd you see this? Well, remember we started the chapter off saying we had red flowers crossed to white flowers, and we got pink flowers. So the pink is an example of a red, which is dominant, that is incompletely dominant. So the reason why it's pink is because the white is showing through a little bit. The red is not able to completely hide the white. So really what Darwin was seeing was a pattern of inheritance. But we didn't discover this pattern of inheritance until some 40 years after Mendel's published work. And we see this a lot. So this is what's happening in snapdragons, right? So the problem is, if you're starting with incomplete dominance, you're probably going to be pretty confused. It's probably not going to make a lot of sense to you, especially if you're like Darwin and you're a blender, right? because first of all, your hypothesis was wrong from the get-go. And you've got a confusing, complicated system because you didn't use reductionism to break it down to the simplest form. So you almost have no hope whatsoever at success. I mean, unless you're just ridiculously lucky, right? And that usually doesn't happen. So let's take a look at what incomplete dominance actually looks like by doing our familiar cross. So in this particular case, I'm going to blow this up a little bit. Here's our cross. Now, this is using 4 clock plants. And a couple things, first of all, notation-wise. So historically, there's a couple of different notational systems that geneticists use, and oftentimes they vary depending on what organism they're working with, right? So for instance, plant people will use one notation convention. Fly people will use a slightly different convention. Yeast people will use a slightly different convention. It's just kind of the way they develop their own mechanism, their own system. So this is one particular type of notation system. It's the exact same thing that we saw before with our upper and lowercase letter convention. Okay, that's the most traditional one, by the way. The uppercase, lowercase letter one, that's the most common one. But like for instance, in botanists, oftentimes will use a slightly different form. We also do this sometimes in humans as well, where we have a stock or a root term like C, capital C, which stands for color, right? That's what C stands for. And then what we do is in the superscript, we just basically say, oh, well, there's different versions of this, right? And this is helpful. Oftentimes you see this kind of notation system if we have a particular gene that has like a lot of different alleles to it, right? One of the reasons why we tend to use this a lot in research is because whenever we generate like a mutation in this gene, you can generate potentially an infinite number of mutations, right? And you may have like a half dozen to a dozen mutations that you're studying at a time of the same gene. It's just mutated differently in each one of these cases. And so you want to differentiate, oh, this is a mutation over here, and this is a mutation that's here, and this is a mutation that's here. And so to differentiate those, oftentimes what'll happen is you'll do this notation style. You'll do like a base for whatever your phenotype is, in this case, color. And then you'll superscript whatever mutation that is. Like if we have 12 different mutations, then we might do like, 1 through 12, right? So we know which mutation we're looking at. In this case, we're using letters to denote the color, the actual color of the allele. So R obviously is red, and then W is white. And so when you cross this, so here you have a true breeder for red and a true breeder for white. So you can see they only have that white and red allele. When you cross them, they're each going to get a copy of red and white, which in an incomplete dominant is pink, right? Because the dominant does not mask completely the recessive. So you get that recessive bleed through. Now let's do the same thing Mendel did. Notice we haven't done anything different than Mendel, right? At this point, we did true breeding strains, just like Mendel did. We got F1 generation, just like Mendel did. Only in this case, instead of seeing one parent, we saw an intermediate, right, phenotype. So this is an example of one of those individuals who did their Mendel, they just recapitulated Mendel in their system. And they're like, whoa, wait a minute, I totally didn't get what Mendel got, right? So what's going on here? As it turns out, instead of crapping on Mendel, they decided to try to figure out what's happening in their system. And because of that, we have incomplete dominance. So let's do the same cross, right? The F1 to the F1, pink to pink. Well, we're going to get the same Punnett square, right? So you basically have a heterozygote. So this individual is going to give one red gamete, one white gamete, same thing with his partner, one red versus one white. When you pair them together, red and red is going to give you a red flower, homozygous for red, right? A red and a white is going to give you a pink one. Another red and a white is going to give you a pink one. And then, of course, a white and white is going to give you homozygous recessive. Now, notice something. The phenotypic ratio here is what? Is 1 to 2 to 1. 1 red, 2 pink, 1 white. But what's the genotypic ratio? It's the same, right? One homozygous dominant, two heterozygotes, one homozygous recessive. So both the phenotypic and genotypic ratio are 1 to 2 to 1. Is that the same we saw in Mendel's system? What was the phenotypic ratio in a monohybrid cross like this in Mendel? Yeah, it was like the 3 to 1, right? So that was the phenotypic. The genotypic was 1 to 2 to 1, but the phenotypic was 3 to 1. So notice how when you cross it, the ratios that you're seeing, the phenotypic and the genotypic ratios, are actually telling you something about which pattern or which set of rules, right, you're using to segregate. And so that's the reason why we always do these ratios in genetics, because that was the way that we were able to determine whether or not our gene of interest that we were studying is segregating in the same way that Mendel saw. And if the answer is no, then the question is, what's the rule set, right? Because that's basically what we're looking at. We're looking at essentially our patterns of inheritance, right? Which is essentially rules by which genes will segregate. And so we have the Mendel's rules, we have polygenic rules, we have pleiotropic rules, we have multi-allelic rules, incomplete dominance rules, and codominance rules. Okay, so now let's take a look at the ABO blood grouping system, shall we? This is going to be one that is essentially going to be describing both multi-allelism and codominance. So we have 3 different alleles in the ABO system, right? We have the same notation system that we saw in the 4 o'clock plants. Our stock term is I, and I basically stands for immunoglobulin, which is a type of protein. It's an antibody, basically. That's what an immunoglobulin basically is. It's a protein that's involved in the immune system. That's essentially what we're talking about. And you have different forms. So the first allele is IA, right? Big A. So that one there is a dominant. You also have IB, which is also a dominant. And then you have little I, which is a recessive. You have 3 alleles, A, B, and I. That's in the population, but you only have 2 of them. Of course, 2 of them as given to you by your parents. Okay. Now, because you've got 2 dominants, A and B are codominant. That is to say, they share power equally, they are equally expressed. But I is a true recessive, and it behaves as a true recessive. So let's take a look and see how this fleshes out in the ABO blood grouping. So here is our genetics. Now, when we take a look at our blood type, and we have our 4 different blood types, we saw this in the lab, right? So we have A type, B type, AB, and O. And remember, these are glycoproteins with different carbohydrate pieces to these forms. In the A type, the carbohydrate is what's called galactosamine. In the B type, it's galactose. In the AB type, it's got both. It's got galactose and galactosamine. Notice I have as many galactoses, triangles, as galactosamines, circles. Why? Because it's codominant. You see them equally. Okay. And then, of course, the O-type one has nothing. So these basically are these are your phenotypes. So one of the things to always remember is a phenotype can be something you see visibly, like when you look in the mirror. However, you can also consider a phenotype. There's what oftentimes when we kind of get into sort of molecular cellular genetics, we oftentimes work with what's called molecular phenotypes, right? So the way you look at a molecular level would be referred to as a molecular phenotype. So your actual galactosamine or galactose, the way you look there, would be referred to as a phenotype, but it'd be like a molecular phenotype because you can't look in the mirror and say like, aha, you're A type, right? So that's a molecular phenotype, but it's still a phenotype. Now let's take a look at the genetics underpinning this, right? Because we looked at that already in lab, but now let's flesh out the genetics. So if you're an A type person, then that means that your genotype can be one of two. You can either be homozygous dominant, that is big A, or you can be heterozygous. Why? Because I is a true recessive. So either one of these would basically give you A type blood, and you would make galactosamine on your red blood cells. If you're a B type person, then again, you would be either homozygous dominant, big B, or you would be heterozygous, big B, little I. Again, little I is classically recessive to B. So A and B are both dominant to little I. Either one of those combinations will give you B type blood. You'd make galactose on your red blood cells. Now, here's where the codominance comes into play. What if you have one of each? What if you're AB type? Well, if you're AB type, that basically means you have one big A allele and one little a allele. You have both. Is one dominant to the other? No. Right? Are they incompletely dominant? No. They're codominant. You see each one of them equally. So if you were to count up the number of galactosamines, you would expect to see a similar number of galactose. They are sharing equally, 50-50 split. Okay, that's codominance. And then, of course, if you're O type, then you are the homozygous recessive. So basically, in order to do blood transfusions, one of first of all, there's two different blood components of your blood type, right? So you have your ABO group, right, which is A, B, and O, but you also have positive, negative, right? And we learned about this in lab, right? The RH factor, which is RHD, there's actually A through E, is also another set of glycoproteins. And in this case, you either have this protein or you don't. So here you'd be positive versus negative. So this is your phenotype. Now, typically speaking, when you're looking at this, the reason why, I mean, think about all the glycoproteins you have on your cell membranes. I mean, there's a lot of them out there, right? The question is, well, why these two, right? So what is it about these two that we have to track? The only reason why we have to track these is because these will essentially create an immune response. So basically, when you take a look at your immune system only recognizes self versus non-self. Not good versus bad. That's it. Remember back when we talked about glycoproteins, we talked about the MHC or self tags that are on all of your cells. That's literally how your immune system works. If it doesn't belong, or if it didn't see this as it was developing, right, then it's going to attack it and kill it. So here's the thing. If you take an A-type person and give them B-type blood, what's going to happen? Yeah, right, because the A-type person is making antibodies against what? B. A B-type person is making antibodies against what? AB person's making antibodies against? They don't have it, right? Because they know both of them. O-type? The whole world's scary for them, right? So they're making antibodies to everybody. So basically, B-type form is a foreigner to the A-type person. Your immune system doesn't see it. They didn't grow up with it, so they're going to attack it and shred it.
This is why it's so important that you have
Yes, because the O-type people will save us all. Correct. So yeah, they have to save themselves, but we all love them because they can save us all. Right? Because if you take a look at it, right, an A-type person can get blood from A-type people, obviously, but O-type people. Why? Because the O-type people don't have anything on their red blood cells that's scary. Right? There's nothing there. So there's nothing to be scared of. Right? So an A-type person can take O-type blood. Depends on the population. Just because O is recessive doesn't mean it's the rarest. And just because the others are dominant doesn't mean they're the most common. Because when we do population genetics, which you kind of talk about a little bit in genetics, but as populations kind of go through their machinations and sort of different cultures and ethnicities and countries, kind of, we tend to sort of herd up in likeness, right? So, and then when we do that, what happens is we start to fix alleles. And so that fixation of alleles is actually more about population genetics as opposed to just pure dominance and recessive. A good example of that is we already have skin tone, hair color, and eye color, right? We already know that the dominant traits for that in humans is dark, right? So dark is dominant for hair and skin, right? Because we're really, really light people are rare. And also eye color, right? That's the dominant version. That's what most people in the world have. If you look across the entire species. But if you go to Scandinavia, that's not what you see at all, right? You see basically a compounding or an accentuation of what essentially is all the recessive traits, right? I mean, light skin, light hair, light eyes. Those are all recessive traits. Just because you see a lot of them in Scandinavia doesn't mean it's dominant. It just means that those alleles in the group that formed that population, the original population that formed those countries and immigrated to those countries, had a richness of those alleles, and they just basically started fixing those alleles. And so they kind of started getting stabilized in the population. It's one of the reasons why population genetics can actually be quite fascinating. And the reason why, when you look at like evolution and how species are migrating and how their genetics is kind of going from one geographical region to another, and how they're spreading genes and how different alleles are getting fixed into different populations. It's a fascinating story when you get into population level evolutionary genetics, because it's really tricky. It's really complicated, but it's like really, really fascinating, because you start to realize that the world looks the way it does because of the way we sort of fixed our alleles in different populations in different corners of the world. Right, so can A give it to B? No, can it give it to AB? Sure can, right? Because AB grew up with A, it knows what A looks like, right? Can it give it to O? No, okay, so it can only receive from A, no. B, same thing, right? Can it receive from B? Obviously, because it is B, right? Can it receive it from O? Yeah, it can receive it from O, because there's nothing scary there for O, right? So the immune system's like, hey, wait, you're just a red blood cell, you're cool, come on in, right? Can it give it to A? Can B donate to A? No. Can it donate to AB? Yes, right? How about AB? Can AB give blood to A? No, right? Because the B will trip A up, right? Can it give it to B? Can it give it to O? No, right? Well, now so it can't donate really to anybody. So if you're AB, you're kind of useless for donation, unless it's another AB person. If you're AB, I'm not saying you're useless, right? But in terms of donation purposes. Now, can AB take A? Can it take B? Can it take O? So AB is what's called the universal receiver. It can take everything because its immune system has been shown everything. So nothing scares it. So it's kind of cool, actually. If you're an AB person and you're hemorrhaging all over the place, you're just like, you know what, just line me up. Just give me one of everything. Just to clear off the shelves. I can take it all, right? But you can only donate to another AB person. Now, the O-type person, can they take A blood? Nope, that's scary. Can they take B blood? That's scary. Can they take AB blood? That's really scary, right? Because you got two of them there. So they can only take O blood. But guess what? Can they give it to AB? Yep. Can they give it to B? Can they give it to A? So these guys are the universal donors. And guess what? Bonfils knows that. So if you're O-type and you see a black van following you all over the city, very slowly, very menacingly looking, and then you wake up in a dark alley somewhere with that missing time phenomenon and the faint taste of cookies in your mouth, it's not alien abduction, it's just Bonfils. They just needed to drain a pint or two. They're tracking you through your fillings, right? They don't microchip you. They just track you through your fillings. So they keep track of you. There's a reason why there's black vans out there, right? That's what they do. So, of course, I'm kidding. They're not quite that bad. However, actually, I've had O-type students, and they're like, you're kind of not that far off. So obviously I'm being a little hyperbolic, but some of my O-type students have been like, yeah, they are a little aggressive. If they know if you donate and you're O-type, they will find you. They will find you, right? So they don't necessarily, follow you around in a black van and kidnap you. But they will put you on every holiday list you want. You want to be on the Christmas list? On it. Hanukkah list? On it. Kwanzaa list? On it. I mean, you want every single list. You make up your own holiday. They're going to put you on it. They're going to make sure that you are well taken care of, and they're going to keep your name on the short list. Why? Because they know full well that the O-type folks save us all. Yep. So O-type people, we love you. So go donate blood. We'll give you as many cookies as you want, but you're going to save the world. Okay. So.
What am I doing? And then they would follow me around.
They will, but for a good cause. They'll be like, you got a lot of blood in there. You don't need all of that. Yeah. So just give us a few gallons of it. I mean, we're good there. You can always make more. I mean, your body's making more all the time. Just, Yeah, they'll give you whatever cookie you want. I mean, that's like, if you want an edible, they're like, why not? Just let us get the blood first, and then you can have as much as you want. I mean, you want a freebase? Here it is. I mean, we got the entire wing over here where you can just have at it. So just don't kill yourself in the process, because we need your blood, right? So that's a thing. Yeah, no, like I said, there's actually an O-type student when I did that first, and they're all like, you're kind of right, actually. They're pretty aggressive about keeping up with you. So yeah, they were laughing. I'm glad they were laughing, because otherwise they're like, oh my gosh. It's like, by the way, if you're A and B and AB, you're good. Nobody's following you, right? So especially if you're AB, I mean, nobody's following you. Yeah. So that's okay, right? So so now let's take a look at a couple of other things. Now, here we have kind of the confluence of, and this is not so much a pattern of inheritance in the truest sense, right? Because the others were basically genetic rules by which genes segregated, right? So clear cut. And they created some sort of a phenotype. Now, this one is not as clean cut as that, because you're not talking about a hard and fast genetic rule per se. You're talking kind of a little bit about a bit of a convolution. This is basically where we get into genetics plus environment equals phenotype. Right now, when we start to get into genetics and environment, getting to your phenotype, this gets sticky, doesn't it? It starts to get a little tricky, because let's say that your genetics was polygenic. What happens if you had like lots of different genes involved in a particular trait, and there's a heavy environmental influence as well? When you have this, like if it's polygenic, we refer to this oftentimes as multifactorial. Multifactorial is oftentimes a term that we use when we're dealing with an incredibly complicated phenotype. that clearly has some sort of a heritable factor to it. But there's also a lot of other stuff in there. A good example, human behavior, right? Like the genetics of human behavior. We know that in lots of human behaviors, there is a genetic component because we can see familial patterns of behavior. But we also know that there is a titanic amount of environmental and social and societal influence in behavior as well. So it's an incredibly tangled gamish of lots of different things that creates ultimately the final behavior that we see. That's what's referred to as multifactorial. So like the genetics, psychology, right? The heritable factors of psychology oftentimes are multifactorial in nature. This one's not going to get so complicated. We're just going to keep it straight and simple as possible, but we're going to take a look at environmental influence. So here's a situation where we have a nice little cat. I'm not a cat person, but I have to admit, Siamese are pretty cool. They got some pretty stunning coloration on them. Those blue eyes are hypnotic. So if I had a cat just to look at, because I wouldn't want it around me, this would be it, right? Because it's really stunning in terms of visualization. So let's take a look at actually what we're looking at here. The way the cat actually gets its pigmentation is through a combination of things. First of all, an enzyme, tyrosinase, which is in the pathway of an enzyme that I study, is at the top of the melanin production pathway. So the melanin, your skin pigment. And so tyrosine, the amino acid, will be converted to another molecule by tyrosinase into what will essentially become the precursors for melanin. Okay. Now that's controlled by a gene, right? That's where your genetics comes from. However, you make the protein with your genetics, but the protein isn't activated until you add the environment. Here's how this works. Tyrosinase activity, which is required for melanin production, if it is in temperatures that are above 33 degrees C. Now, just to give you a sense of reference, 37 degrees C is our core body temperature. So if it's above 33 degrees C, so warm, right, then tyrosine is inactive. That means you're not going to be making melanin. As a result, in those areas of the body, you don't have a lot of pigment deposition. So the result is lighter colored fur. That's the creamy sections of the Siamese cat. In areas that are below or cool areas, so this is hot, and these are the cool areas, then if it's below 33 degrees C, tyrosinase becomes active, and then you get deposition and creation of melanin. And then that results in these dark areas. So like, for instance, the face, which is deposition of pigment. Now notice where you see this. The hot areas of the body, the core, are too hot for tyrosine production or tyrosinase production or activation, and therefore are lighter in color. The cool areas, the extremities, the things that get cold on us, right? The things we tend to sort of bundle up. You wear gloves. Why? Because your hands get colder than your core, right? So these areas, the extremities, are cooler. So you get active tyrosinase activity, melanin production, and deposition. So these are the areas where you see dark. So you can see the actual phenotype of the Siamese cat is a confluence of the genetics of tyrosinase production plus environmentally cued tyrosinase activation. Yeah.
How, like when you see the above and below, like how does it have a difference?
So that would depend on the kinetics of the enzyme itself, which I have no knowledge of because I study the enzyme that's in the step before tyrosinase. So I've never looked at the actual kinetics of tyrosinase. Well, my guess is it's probably not a lot because you usually don't, because you don't want to have big wild oscillations of body core temperature because then you're going to get hypothermic or hyperthermic. Those are bad scenarios, and you do that very quickly. within just a few degrees. I mean, we know that, right? Because we know that if we go from 90.6 to like 100, then that's like an illness, a pathological state of increase in core body temperature. So you know it can't be a lot. So you're probably talking within a few degrees or so. So this is probably a fairly sensitive enzyme. So what would happen if during development you put like a heat pad on this cat's tail? Yeah, it'd be creamy, right? You'd expect it to be kind of creamy, maybe. You know, kind of depending if the heat kind of got penetrated, because what we're talking about here is core body temperature, so it's from the inside out. So it had to be from the outside inside to actually get into the core area. But this is actually kind of an important sort of a thing, because when we take a look at this, you can see how your phenotype for the cat is not just all genetics, right? It's also environmental as well. And this is kind of an important thing, because we see actually, when we first saw this, we were thinking like, oh, well, this is an outlier. Most of it, like 90% of it is genetics. And then there's maybe a little bit of environmental stuff here and there, just to kind of like a seasoning on a good dish, right? Most of it, the dishes are there. The seasoning is just kind of a little bit, you know, it's an outlier, right? We actually, as we got deeper and deeper into genetics, we realized that this whole non-genetic thing is actually a bigger deal than we first thought. That there's a lot more in there, that this is a broader area than we had originally thought. And it has titanic consequences on how the phenotype comes out. Let's think about this for a second. How could this possibly be advantageous? Now, the cat's a bad example, but you know, think of, say, for instance, oh my gosh, what rabbit is it? Is it a Himalayan or something like that? It's kind of white, but it's got like black ears and like a little black nose. It's not the snowshoe, because it's all white. I think it might be the Himalayan. Right. So the question is, now, the reason why I want to switch to the Himalayan is because rabbits basically are in everybody's menu. Okay, so you're at the top of the menu for most carnivores, which pretty much everybody you meet wants to eat you. You're not paranoid. It's actually true. We are out to get you, right? Which is the reason why rabbits and prey species act the way they do. Like you want to eat them, like all the time, because that's mostly true. But think about it. You're this like a little Himalayan out in the snowbank. You got these black ears, right? And this like little black face. And the question is, how in the world would that be a good idea in the middle of a snowbank when you are at the top of everybody's menu? I mean, isn't that like putting a big sign on your head saying, eat me? Right? I mean, doesn't it seem like the snowshoe hare has it right? Right? Just kind of disappear into the snow. So how is it that the Himalayan has the same basic idea for the Himalayan, actually? Right? How is that a good idea? Well, think about it. All you care about is living, yes? So when the predator starts to chase you, you've got some advantages. Number one, you're mostly white, but the areas where you're black are strategically placed. Think about it. If this were a rabbit, or if I wanted to kill this cat and eat it, right, let's say, for instance, what would I go for first? What I can see the best, right, which is the dark areas. For the Himalayan, it's the dark areas. What am I going for? I'm going for the ears, because I can see it. What am I not going for? The kill shot, right? The kill shot, which is generally the core, because I can't see it so well. So even if I do get a hold of it, I'm likely going to get a mouthful of ear, which, you know, whatever, take an ear, I'm gone, right? So strategically, the color placement in the Himalayan is designed to confuse the predator, number one, because it sees just a sort of disembodied black blobs bouncing around in the snow. And then you attack the black blobs, which means you're off target. So you're less likely to get your kill. So what you're doing is you're kind of decoying it, right? You're saying, here, don't you want to attack this over here? Attack that over there. You can have that, right? Squirrels do a similar thing with their tail, right? That big old fluffy bushy thing. What's the first thing you notice about a squirrel? That big old fluffy tail, and they know how to work it, don't they? I mean, they're all like, I mean, they got that thing vibrating all over the place, and when they're threatened, it's like all up in the S shape, and they're like waving it around. Why? Because what does a predator grab first? The tail, right? Does the tail matter? No, I can tell you not, because actually we have quite a few squirrels around our house who have half a tail. It's like take it, whatever you want, take it, right? So You can have whatever you want, right? Same idea. It's a decoy. So it's a strategy sort of thing. So basically in this case, I'm either grabbing the tail of the cat, the foot of the cat, who cares, right? The ears of the cat, but I'm not going for the kill shot. So strategically, there's actually a strategy associated with the actual pigment deposition. So it's there for a reason. Okay. So the environmental thing was, yeah, I'm not sure where that fits in here, but let's just add it. I'm just going to add it just as a placeholder, because the other one that I want to look at is epistasis. Epistasis is basically a violation of D. This is a situation, this is where one gene affects the expression of another. Expression and segregation, oftentimes. We don't talk about a lot here in this chapter, but this idea of non-randomness has a term to it that we look into a lot more in depth in the genetics class. We call it linkage. There are some genes that are a package deal, that what happens in A does affect what happens in B, that A and B go together, that it's like a package deal. So they're not randomly segregating. So we do see that non-random segregation. We call that linkage. So that would be a violation of Mendel. We get into that a little bit more in detail because it's actually pretty fascinating how that works out. But epistasis is mostly focused on expression of a gene being affected by another. So this is the best, since we just talked about pigment. Now, Emerson worked on corn, and we'll talk about corn. And so what he did basically was Mendel's work, but instead of seeing 9331, what he saw was 97. Right? Notice what 97 is. It's a variation of 9331. Right? So if you take 9331 and you add these together, then you'll get 97. That basically means he did a dihybrid cross. But guess what? Those two genes were not random. They were talking to each other, right? So what was happening in one affected what happened in the other. So that's what epistasis is. Now, what I want to do is I want to use the most basic example, and then we'll take a look at Emerson's corn work. So I want to look at the most, what we just looked at. So imagine we have a pigment gene. And we have two alleles in it. So big B will make black pigment. Little b will make brown pigment. So that's one gene, right? That's my pigment gene. However, there's another gene, an epistasis gene. We're going to call it E. In this particular case, big E is basically saying, make pigment. Little e is saying, don't make pigment. So you can see what happens is you have a kind of a decision tree, don't you? Where one decision is reliant on a previous decision. So if I basically take an individual that looks like this, and the epistatic gene looks like that. What color will that be? So this will make black pigment. If I have this, what color will this one be? This will make brown pigment. But what if I have this? So this one will make no pigment, which typically will be white, right? Because the decision in E has to be made first. And this one said, okay, since I'm dominant, right? I've got, dominant recessive. Since I'm dominant, I'm saying go ahead and make pigment. That tells B, do what you do. You're going to make black, so make black, right? Same thing. If I've got two of these, I'm dominant. So that tells the B gene, do what you do, make pigment. So it makes brown. But if you are homozygous recessive, then what you're saying is don't make pigment. Guess what? Doesn't matter what B has because you've been told not to make pigment. If you had been told to make pigment, what color would you have made? You would have made black, but you were told not to make it. So you make no pigment, which typically comes across as white. So then you have three different colors. So you can see then that the expression of B is reliant on the permission of E. Make sense? That one permits the other. That's epistasis. And so what Emerson did was basically cross corn. And so he was sitting there looking at two different genes. And so he took some compound heterozygotes that were purple. And then he crossed them together and he got the same Punnett square that we normally get. Only in this case, what he noticed was that when you have a dominant in both, you're purple. Dominant in both, you're purple. Dominant in both, you're purple. That's true for all of these. But when you're dominant in A, but recessive in B, then that seems to suggest that you can't make pigment at all. So what that means is that B is epistatic to A. Right? Because B just said, listen, you're not making pigment at all. Don't care what color you are. You can be fluorescent yellow. You're not making pigment at all. So what color are you? White. So that basically means that B is telling A, no go. Right? Then of course you get two dominants, color, and now you have, again, a dominant in A, a recessive in B. Again, same thing. B is basically saying, listen, sorry, A, you're not going anywhere. You get two dominants in purple, two dominants in purple. Now you have a recessive in A and a dominant in B. But guess what you get? Again, you get white. So what this is telling you is that little a is acting epistatically to B. So little a is saying, hey, guess what, B? When I'm little, you're no go. So if I'm not here, you're not here, right? Same thing here. Again, nothing there. Get two dominants, you get color. You get one recessive and it represses the other. The recessive A represses the B. And then of course, if you have both of them, there's nothing going on there, right? So this is a little bit different, right? So essentially, when you take a look at the 9-7 ratio, what it's saying is that you have two genes that are actually acting epistatically on each other. Which, what does that mean? Which means both are required for expression. So you have to have both dominants in order to get your color expression. If you don't have both, it's not enough. Now, what we later learned on is that this is actually a biochemical pathway. It makes total sense, right? So here's basically a colorless precursor, which is converted into an intermediate that is also colorless by A, right? That's your A gene. So it's an enzyme that's encoded by the A gene. And then the B enzyme encoded by the B gene will turn this intermediate into your purple pigment. Well, here's the problem. If you don't have any B, then you can make as much colorless intermediate as you want. It's never going to get past this last step. So that basically means that the presence of B is required to get purple. If you were to miss A, then you would actually never get your colorless intermediate. So B would have nothing to work on. So that basically means that A is also required in order for us to go through. So basically, either one of them acts epistatically on the other. They need each other in order to be able to generate your phenotype. So that's a type of epistasis. So for you table people, here's your table of all your different things. We have polygenic, pleiotropy, multiallelism, dominance with codominance and incomplete dominance all together in one place. Environmental factors, right? That's our Siamese cat and our Himalayan bunnies. And then of course, gene interaction, which is the epistasis piece of it. And there's a lot of other gene interactions. Epistasis is just one type of gene interaction where two genes interact with each other in a non-random way and kind of create all these crazy ratios. There's a lot of other gene interactions that we look at in the genetics class because this genes are much more fascinating and complicated than just what Mendel saw. He just kind of got the ball rolling. But in reality, there's a lot going on in there. Okay, let's go ahead and dig into replication, shall we? Okay, I'll be right back. I got to get my phone because it's sending me a push to it. So now, so that's kind of a big sort of topical break, right? So that's all genetics. And I know that you guys will be eager to take it in the fall. Or not. Okay. So chapter 14 is basically 14 and 15 is basically replication in 14 and then transcription translation in 15. So where we are now is kind of, now that we talked about genetics and all that sort of stuff, now we're talking, because we talked a little about cell cycle, we talked about G1, S, G2. And we said, okay, S phase is basically replicate, right? Duplicate and separate. Well, this is the replication chapter. All right, so now we kind of get to the nitty gritty. So we kind of put that one on hold for a second just to kind of finish out the cell cycle and genetics component of it. Now we're ready to go back and take a look at the actual events that's happening in S phase, and that's basically replication. But we all start with essentially understanding DNA. And understanding DNA with respect to its role as the genetic material. Now, this kind of sounds like a yeah, duh moment, right? Because we basically spend the majority of the biology semester drumming it into your head that everything is that you need for information wise to build you is in your DNA, right? So we kind of basically say this is your genetic material. We've been talking about how You're passing your chromosomes on from one generation to the next by all these rules we call patterns of inheritance and genetics that create your phenotype. So it's kind of like we've been drumming it into you. But the one thing we don't really mention is that in reality, we didn't really know that this was a genetic material for a long time. Because remember, Mendel, when he did his work, didn't actually know what genes were. He didn't know what chromosomes were. He didn't know anything about mitosis or meiosis. He didn't know about cell cycle. He certainly didn't know about checkpoints. He didn't know what alleles were. He didn't know any of that stuff. So what was Mendel actually doing there? What Mendel was following was information. All he knew was that the little gold dust that he put on his paintbrush from the male donor has some sort of a factor in it, some information in it that is essentially the instructions for purpleness. And that when he rubbed that onto the stigma of the female, that the female also in there somewhere had the instructions for white. What that was exactly, no clue. He just knew that the information or the instructions for purple came from one and the instructions for white came from the other, right? That was the segregation. That was the law of segregation. The idea that it was segregating pollen to whatever was in there, right? And so didn't know anything about DNA or proteins or carbohydrates or any of that stuff. So really, when you take a look at what Mendel did, it was actually pretty amazing what he was able to actually accomplish, considering he knew nothing, literally nothing about chapters 10, 11, and 12, right? So that's actually pretty amazing that he was able to put all that together. But however, so what's the glaring question coming out of Mendel? It's obvious, right? So what is this particle? So in terms of Mendel wouldn't have thought of it as a gene because the term gene came later, right? So what Mendel would have thought of it is like, what is this? So you have some sort of a particle, right, of instructions. What is this particle? What is it? Well, we don't know, right? At least at Mendel's point, we didn't know. We didn't know actually for a good long time. As a matter of fact, the first half of the 20th century was working on trying to figure that out. Our first stop is with a classic experiment by Frederick Griffith, which happened in the Roaring 20s. Actually, if you look at 28, just to kind of give you an idea, America-wise, what was happening in 1928. 1928 was the end of the Roaring 20s, right? So if you think Great Gatsby, if you want to read a good novel about the 20s, The Great Gatsby is probably a good one. But in 1928, that was the end of Calvin Coolidge, his presidency, and Herbert Hoover was elected. Of course, we all know what happened in 1929. Yes? What happened in 1929? You're like, 1929 happened? What? Well, one was in the teens. We were kind of in the interlude. The First World War started in, well, it depends on where you want to put that. I mean, Hitler was elected chancellor in 33. And then it got ugly in the late 30s. And then it came to America, obviously, in 41. So it's kind of a little progressive. Yes, right? The beginning of the Depression is what happened in 29, the stock market crash. So America-wise, just to give you a sense of context, this was the end of the Roaring 20s. At this point, we didn't know it was the end. It was good times. That's the reason why Hoover was elected, because he was like a good time Charlie kind of a guy. I mean, he was like a business guy, and it was like, hey, let's just keep the dice on rolling, right? And then, of course, the stock market crashed in 29, and then that's what basically laid down the 30s. But in this time frame, Frederick Griffith did a landmark experiment, and basically what he was studying was strep. Actually, the Streptococcus is actually a genus name. There's several different species under the Streptococcus umbrella. One particular one, pneumoniae, is the one that actually causes pneumonia. Okay, it's not the strep of strep throat fame. That's a different species of Streptococcus. Okay. So he was looking at, now notice the time frame, right? Mendel published in 1865. His work was largely forgotten it was rediscovered in the early 1900s. That's when William Bateson and William Punnett and others dug it up, and they started doing like work. They tried to do Mendel's work in like 4 o'clock plants and corn and stuff like that, right? So that's happening about 20 years before this. So a lot of folks had already been doing a lot of this genetic stuff in their own systems, and they've been trying to sort it all out, trying to figure it all out. What was also happening, we talk about 4 o'clock plants, we talk about corn plants, we talk about fruit flies. That's the one we use actually in the genetics class, is fruit flies, but along with molecular stuff. But there was also a group of people working on humans as well, and Frederick Griffith was one of them. He was trying to figure out human pathology. In this particular case, he was working on pneumonia, because back in those days, pneumonia was a bad deal, right? This was pre-antibiotics. So you didn't have antibiotics at this point. So if you got pneumonia, it was just largely bundle up, stay warm, and pray. That's pretty much your therapeutic approach, right? And so the idea here was, well, how can we get the upper hand on this one, right? How can we understand the system and try to get the upper hand on this one so that we can sort of beat this one? Because people die of pneumonia a lot. We still die of pneumonia a lot. And pneumonia is actually a pretty difficult one to deal with, because it gets into the lungs, and when things get into the lungs, it's difficult to manage. So here's what he had. He had 2 different strains of Streptococcus. So he was looking at. One strain was called the S strain. This was the virulent strain, right? So that basically means it causes disease. That's what virulence means. And of course, if you get disease, pneumonia, you die. That's the reason why I have the little frowny face. Right? Now, the R strain, he noticed, was another strain that he had, but this was non-virulent. So this one was not causing disease. Naturally, being a curious scientist, he wanted to understand a little bit more about how this worked. Because the idea is, maybe we can use this to our advantage to try to beat this strep and kind of give us a bit of an upper hand on pneumonia. Notice he's not at the point where he's like, biomedical research. He's not like trying to develop a cure for strep right now, for pneumonia right now. He's just trying to understand the system better. That's where all biomedical research starts. All biomedical research starts with basic science, right? Because you can't understand how to cure a disease unless you understand just the fundamentals of how it is supposed to work in the first place, right? So that's kind of where he is right now. He's just trying to understand, how does this all work? And then maybe we can use some of that knowledge to our advantage. That's what he's still looking for. So what he noticed, and he was working with mice, so not a good place to be one of Frederick Griffith's mice. So what he noticed basically was, he took these mice and he infected them with these different strains. And here's what he got. So if he infected his mice with the live S strain, then he noticed that this was a situation where the mice died, which makes sense, right? Because the S strain basically causes disease, the mice get pneumonia and they die. If he infected them with the R strain, in this case, your mice live. So that's a good thing. That's what we want, right? Now, here's what he did. He just want to leave it there. He said, well, wait. Let me think about this for a second. So we've got this potential bacteria rife. I mean, think about hospitals back in those days. I mean, we were still a little loosey-goosey on sterile technique. We were probably pretty horrible at sterilization. Hospitals were probably tragically dirty places because it was just capitals, meccas of disease. And hospitals are still that way, by the way, right? I mean, you don't see a staph infection anywhere in normal life, generally, unless you go to a hospital. Right. So that's just where you are. It's where we go. We get sick. We leave all of our germs there. That's just what happens. Right. So you can imagine that hospitals in this era were probably pretty dangerous places to go. Right. So here's the thing, right? The thing is like, well, wait a minute. If this bacteria is all over the place because we've got our pneumonia wing and we're going to keep them separate, and you've got these pneumonia bacteria all over the place getting everybody else sick. We got a little pandemic going on our hands here. How do you beat it? Well, one of the things he wanted to say, one of the proposals was, well, maybe we can sterilize things by heat killing it, right? So basically just this same thing we do in the mountains, right? We boil our water. Why? We're trying to heat kill the Giardia so we don't get rampant diarrhea, which is never a good thing. especially if you're up in the mountains. Just saying, right? That's not a place you want to be, because you're already probably a little dehydrated. That's going to make it worse. Good luck getting down. But anyway, or at least getting down in any sort of a presentable form. But anyway, I'll leave that to your imagination, right? So the idea was this. This is the idea that was happening in clinical environments back in those days. It's like, well, wait a minute. What if we take our instruments and we just, just either really just steam them or heat them or just like burn them, basically, right? I mean, we kind of do the same thing with a flame. We kind of like flame some sort of a needle or something like that to sterilize it. What are we doing? We're heat killing any bacteria that's on there. So there's an idea there. So it's like, well, what if we heat kill this S strain and then infect the mice? Well, in this case, what happened was he got happy Mickeys. Great. It's looking good now, isn't it? Maybe the strategy is tell everybody in the clinics to heat treat all of your utensils. That'll reduce and kill all the bacteria and we're good. But guess what? When you heat treat it, you don't have a way to remove it, do you? You're just killing it and you're leaving the little bacterial bodies there. So here's his next question. Well, if we're not really removing it and the live strain is still there, right, the R strain, then what happens if these two come together, right? What I want to do is I want to mimic what would actually happen in a clinic. So I heat treat a utensil that may have S strain on it, but there's R strain all over the place. What happens if the R strain gets in contact with this utensil? And now you've got dead S strain and live R strain on there, right? So that's what he did. So he said, I'm going to take the heat killed S strain and the R strain, and I'm going to infect the mice with it. Interestingly, what he saw was dead mice. Wow, all of a sudden you just hit the brakes, didn't you? It's like, wait a minute, maybe heat killing your utensils isn't going to work as well as we thought. Because it looks like if we have any live R strain, if it comes in contact with the dead bodies of the S strains, it's still going to be a problem. So what does that mean? That means you can't just heat kill it, can you? You actually have to remove it. You have to get rid of it somehow. It has to be gone. So this is kind of what it looks like in pretty little pictures. So here you can see you take basically the S strain, you inject the mice with it and they croak. Take the R strain, you inject the mice with it and they live. So very nice. Right? So when you take a look at a bacteria like this, typically it's associated with the coat, that polysaccharide coat that they've got in this particular case. When you have the heat killed S strain and you inject in the mice, they live. Happy mouse, right? But then when you add the two together, heat killed S strain, dead body, basically, plus living R strain together gives you a dead mouse. Why? Well, this was fascinating, right? Basically, what Griffith noticed was that there was this process by which the S strain cells were actually able to actually transfer the information. So if you just want to stick with Mendel's work, right? Because at this point, you don't know the DNA is the thing. But basically, what is the information for virulence is transferred from S to R. So what is this telling you? Without knowing anything about which molecules is the culprit here, because you didn't know it at this point, right? What you're basically saying is that the information for virulence, what causes virulence, whatever that is actually being transported somehow from the dead S cells to the R cells. So what he's saying is there's a mechanism for the DNA to physically move. In this case, he didn't know it was DNA, right? But the information to physically move from one cell to the other. That then turned the R cells virulent. So once the R cells get infected by that virulence factor, then they become virulent themselves. That makes sense? So this basically transformed R cells into virulent cells. So even though you killed the cell, you didn't destroy the information. That's what this says. So what Griffith stumbled on is a process we refer to as transformation. There's two different terms for transformation. One transformation is a cancer term. It's the transformation of a tumor from a benign to a malignant state. It transforms, so it changes its identity. But at this level, transformation is associated with the movement or the transferal of DNA from one to another. This is the first time we saw this, right? So basically what we're saying is that you can actually move cells from one to the other. And that's something we noticed in bacteria. So that's a bit of a head scratcher. Because now all of a sudden we notice it's like whatever that is, if we can find whatever that is, it's moving from S to R, then we're going to find that inheritance molecule, whatever that particle is, right? But the question still remains, doesn't it, right? What is this particle? We still don't know. Well, if you kind of fast forward to the 40s or so, again, at this point, World War II was raging now at this point. It was almost over, at least for us. The worst part actually was just beginning for Europe for the most part. But for us, we ended in 45. So we were still at war at this particular point. And so what Avery, McLeod, and McCarty did was they did Griffith's experiment. And they purified the cell extract. So they took the components of the cell and they kind of did like a cell, like a gamish of cell, like cell stuff, cell guts, basically, because they were looking for what that was, right? What is inside the cell that's causing this to happen? They wanted to know what that was. So what they did is they basically took cell extracts, which is a fancy way to say cell guts. And then at this point, we kind of started having some ideas about what the major macromolecules were, right? We already knew that proteins were a thing. We knew that nucleic acids were a thing. We knew of it, right? We already knew that carbohydrates were there and lipids were there. So we already knew that these were the four major macromolecules. The problem is we didn't know which one was the inheritance molecule. There's a big debate going on as to whether or not it was protein or nucleic acid. And so what Avery, McLeod, and McCarty did was they basically took these cell extracts, they sucked all the protein out of it. And then they tried to see if they could use these cell extracts, because the idea is like whatever was inside those dead S cells is still there. It's just that if you just sort of like ripped it all open into like a big slurry, right? So it should all be there. Whatever that is, that should still be there. So what they did is they subtracted out the proteins. So the proteins aren't there. And then they fed that to the R cells. And what did they find? You didn't destroy the transformation ability. So the R cells still transformed. So what does that mean? What's your conclusion from this experiment? Is protein the genetic material? It's not protein. Oh, but wait. But there's a problem with this, right? They didn't know about this yet. But there's a problem with this, right? Because DNA isn't just DNA, is it? DNA exists in complex with what? Proteins, right? All those histones that they're wrapped around, those are proteins too. So chances are you probably weren't able at this point to actually get rid of all the proteins. So there's still probably some proteins left behind because probably the DNA is still wrapped around those histones. So there's probably still some proteins in there. Okay. So then to kind of combat this question, it's like, well, maybe you didn't get all the proteins. How do you know? Did you test to make sure you got all the proteins out? Think about how many proteins you're talking about. Is it possible you left a few behind? Right? I mean, you can imagine all the questions, all the pushback questions, like, there's some issues. I got questions here, right? I don't necessarily believe your results. Your conclusion is it's not protein, but what if, right? Now, what they did then was a partner experiment where they took a look at the DNA piece of it. So they said, okay, fine. We tried to remove the proteins and show you, but now let's do the flip of it. Now let's remove the new DNA. And then let's see what happens. So what they did is they used restriction enzymes. Remember those restriction enzymes we talked about in the electrophoresis lab? They used those digestive enzymes, those nucleases, to chop up the DNA. So the question, so they basically the idea is this: if it's DNA, those if-then statements, that's a prediction we're making here, right? If the inheritance particle is DNA, then when I chop it all up into little itty bitty pieces, then that genetic information should be gone. True? Okay, so what did they do? They destroyed all the DNA. And then what did they see? They destroyed that transformation ability. So the R strains did not transform. What's their conclusion there? The DNA is the information molecule. Megan. They are, because they have the digestion enzymes. They don't really understand it that well, right? Because at this point, this is pre-DNA structure. So our concept of DNA is a little fuzzier. We know that there's nucleic acids. We know that there's two different types of nucleic acids. We know that DNA is one of them. We know that there's RNA. We don't understand RNA very well at all at this point. And we don't have any clue whatsoever about mitochondrial DNA, right? So our conception of DNA is hazy at best in this particular situation. By a lot. Yeah, by a couple of decades, at least. It's not that we didn't know that RNA was a thing. We knew that there was different types of nucleic acids. We just didn't really understand what it was, how they differed, or what they were doing, much less their structure. So there was a lot of work that we had to do there because most people were interested in DNA. And so RNA didn't get a lot of love, actually. So this sounds pretty compelling, right? Because both these together basically supports DNA as your genetic material. But were we happy? No, of course not, right? Because we got questions with this one too, right? Think about it. Well, you know, our understanding of DNA is a little hazy at best. So how do you know that that you got all the DNA? Did you test for that? Can you prove to me that you have no DNA in there at all? Right? What if a little bit was left over? Right? What about other nucleic acids? Did those get digested? Right? I mean, so you can see all sorts of questions because there's lots of questions now, not a lot of answers. So this was the beginning. Now, in hindsight, we look at this and we think, well, this should have been it, right? You should have been convinced after this one. But this was really the first experiment to really significantly suggest that DNA was a genetic material. Generally speaking, it's a process, right? Usually we don't just do an experiment and say like, aha, I got the answer. Let's all just sign on. That's not the way it works. Usually it's like, I've got the answer. And most everybody says, no, you don't. I've got lots of questions. I don't believe you yet. And that period lasts for potentially a few decades. As slowly over time, other people do what you did. And they say like, you know what? The guy over here said he got the answer. I'm getting the same answer. And the more people that convince themselves that that answer is what it is, and the more they do experiments that are consistent with your answer, then slowly over time, and I do mean slowly, we eventually sign on and say like, yeah, at this point, I think we're all pretty comfortable saying that's the answer. Which is one of the reasons why, if you've ever wondered, why when people get the Nobel Prize, they get it for stuff that they started like potentially decades ago. Because it takes that long for us to really finally get to the place where like, okay, yeah, you know, I think we're willing to admit that after a few decades of work that your discovery of green fluorescent protein was pretty significant. So we'll give you the Nobel Prize. But there's a process to all that, right? And it's a long process. It's a process of intellectual evolution. And sometimes you have to sort of change minds. And honestly, let's face it, we are stubborn pigs. Nobody. First of all, lots of egos. Research is driven by people who are used to being the smartest kid in the class. And they're used to being right. And that basically fosters massive egos. So when you say you've got the answer, what you're really saying is that, oh, you don't have the answer. Okay, so that's going to take some time for a big ego to get around, right? And there's a lot of big egos in science. Most big egos, the response is, no, you don't. They push back on it, right? It's like, I don't believe you. Prove it to me, right? Did you check for this? Did you check for this? Are you sure? Do you think you could be wrong? Because if you're right, then that means I'm wrong. And I don't think my frail little ego can handle that, right? There's a lot of ego in there. And that's the reason why it takes so long, is because we're just slow-witted humans. Gifted in science, but socially and emotionally stunted, for the most part. That describes a lot of researchers. They are socially and emotionally stunted to the point of almost dysfunction. So. Huh? Yes, some of them might be British, but I think most of them are American. Yeah. Like Avery McLeod McCarty, I believe they were British, or at least one of them was. At this point, we're kind of going back and forth and collaborating. But these guys, I think, are mostly American, or they're Brits who are in America, because at this particular point, I mean, I don't know if you've seen pictures of what London looked like in 1944. I mean, this is like the right before Hitler did his last gasp, right around in that era. And he just annihilated London. So that's like during the London bombing. So Britain had its hands full at that particular time. So if they were British, they came over here. Now we kind of go over there, right? So to hang out with folks in Oxford and Cambridge and all that kind of nonsense, right? So then following up with Avery McLeod and McCarty came Hershey and Chase. And this one was the lockdown. Now notice you got the beginnings of it, right? With Avery McLeod and McCarty, which was a big deal. But we wanted something a little bit more discreet, right? Because the idea was like, okay, fine. So, but here's the problem with using cell extracts. You didn't really know what the particle was to begin with. When you do cell extracts, which is basically cell guts, you don't really know what's in there. So there's a lot of unknown variables in there. And I mean, we just don't feel good about this, because you're just talking about a slurry of a bunch of stuff. And most of it, we have no clue what's in there. So is it possible that there could be sort of like some magic black box molecule that we haven't discovered yet in that slurry that could be causing all of this? And this is all artifact. And the answer is yes, right? At that point, if you don't understand the system very well, then you have to understand that there could be something in there that we don't know about yet that could be causing all of this. And that's kind of where the pushback was on Avery McLeod McCarty, because it was good, but it wasn't rigorous enough and definitive enough to eliminate some of those questions, right? There's just too many variables in a slurry. You've got the entire cell's worth of stuff, proteins, everything. And so you haven't really systematically gone through and eliminated all of those as possibilities. Okay. So that's the reason why people were not as satisfied with Avery McLeod McCarty. They were curious. It piqued the interest. It kind of whetted the appetite, but we wanted something a little bit more shut down, right? So lock it all down. There's no way to move to the right or the left. This is it. That's what Hershey and Chase did. Hershey and Chase is a landmark. So in 1952, this is a landmark experiment that was happening. Just to kind of give you another, since we're doing pseudo history along with this one, right? In 1952 in America, this is basically deep in the bowels of Europe, is basically trying to reconstruct itself, right? This is kind of where the Marshall Plan is starting to get developed. I don't think it got developed in 52. I think it was within a few years of 52, the rebuilding of Europe and all that sort of stuff. And then this is also an election year for us. This is when Eisenhower got elected. Truman stepped down at this point. So that's kind of what was happening in this area. We were post-World War. The soldiers had come home. The baby boom was on. So the hippies were being born at this time. So as soon as the war ended in 45, up to this point, you are talking about the baby boomers. That's when these guys are being born. Okay. who are all old now and retiring and otherwise going into assisted living and retirement homes. This is the reason why every single freaking apartment building goes up as a senior apartment home, because there's a lot more baby boomers out there than all the rest of us. Right. That's just what happens, apparently. I mean, you go to war, you get shot at for a little while, it kind of sucks a little bit. What do? You come home and you have kids, right? That's kind of your stress release, apparently. But I guess that's the way it works. But so what did Hershey and Chase do? So what Hershey and Chase did was they set up a system that we kind of have to explain a little bit. So they worked with what's called bacteriophage. Now, a bacteriophage is essentially like a virus that infects bacteria. And so when you take a look at bacterium, here's your little bacteria. A bacteriophage is literally composed of only DNA and protein. So if you take a look at a bacteriophage, it basically has like this little protein capsule, and it's got like a little collar piece also made of protein. And then it's got kind of like these little legs. Looks like a little lunar lander or something like that. And then inside, so that's all protein. And then inside is going to be the DNA. Everything else is protein. So the way that bacteriophages work, and by the way, viruses are also similar. So our studies in bacteriophage, 1952, was actually the beginning of virology. As it turns out, viruses, including coronavirus, of which COVID is one, right? There's other coronaviruses, by the way, just to let you know. Ebola, for instance, is a coronavirus. But viruses are the same thing. They're just protein and nucleic acid. Now, most of them are DNA. So here's how bacteriophage works. So what'll happen is when bacteriophage basically come and they infect the DNA, what they do is they land on, or they infect the bacteria, they land on this guy, and then they kind of dock on him. And then what they do is they inject their DNA into the bacteria. What happens at that point is the DNA hijacks the bacteria's systems. So all the systems where the bacteria is using to make its own proteins from its own DNA, the bacteriophage hijacks it and uses the bacteria's systems to make its own viral proteins, which are sometimes little leg pieces, little collar pieces, and little head pieces. And then eventually it assembles them, and then it'll lyse the DNA, the bacteria, and it'll release other virus particles will go and attack other bacteria. So it's kind of like, if you want to think, so we're talking about the Germans, right? It's kind of like the Germans, what the Germans did with the factories in the 30s, right? So you have a baby bottle manufacturing. What they do is they take over the factory, and then they basically say, listen, you guys are going to make us bullets now, right? So what do? They basically invaded, they infected the factory, and then they hijacked the factory. And they said, listen, you're making our stuff now. So you're going to make our grenades and our bullets and stuff like that, right? So basically that factory was infected. And so this basically is what's called, this is the pathology. So this is the phenotype. It's what's called the lytic cycle. So this is the phenotype that you're looking at, this ability to infect and hijack the cell and basically make other viral particles. That's the phenotype they're following. And here's the correlation. The phenotype should follow the genetic molecule. So it looks like we're a little beyond our witching hour. Let's get excited about all this stuff. Okay. And so what we'll do is we'll start with that, and we'll take a look at the Hershey-Chase experiment and how they basically tease this apart. Okay. So I'll meet you guys down in the lab.